Thank you very much for that kind introduction, and thanks, ILSI, for inviting me to take part in this session. I'm going to stand down here to try to be a little more friendly as we talk through some of the things that I'm going to say. For those of us that are in our 4th or 5th decade as toxicologists, we've probably developed our career on single chemicals, the adverse effects of single chemicals, whether they're in food or in drugs or pesticides or whatever. And so that's really a challenge that I would put out to those young folks that are coming into the field to think about where the real issues are, and that's in mixtures, regardless of whether we're talking about foods or any other type of material. So we're going to talk about some of the approaches for mixtures. This is my disclaimer. And the 3 things that we're going to spend our time talking about are, why are we concerned about mixtures? Secondly, we'll talk a little bit about hazard assessment of mixtures, and then we'll get into some examples that we can look at. When we think about mixtures, it's really what we're all being exposed to continuously, whether it's in our food, whether it's in the water that we drink, whether it's in the air that we breathe, or whether it's the cosmetics that we use. Regardless of the source, we're not being exposed to single chemicals. We're being exposed to mixtures. And if we think about that, Superfunds from the standpoint of the Environmental Protection Agency are mixtures, and what you had for lunch today is nothing more than a mixture. And they're complex mixtures. And we need to begin to think about how we're going to handle these complex mixtures. And it's a real challenge that's out there before us. And there are a couple of issues as we begin to think about these complex mixtures. The first of all is, how are we going to approach the problem? Are we going to go the way that we have been going in the past by looking at the whole mixture and then evaluating that in various types of tests? Are we going to begin to break down that mixture and look at the component parts? And then the other area that we need to give consideration to is similarities. How do we handle, and can we cross over, can we read across between mixtures if we have sufficient information about that? So when we think about the human health hazard of mixtures, and we're only talking about hazard, the human health hazard of mixtures, we can look at it from the standpoint of the whole mixture. Again, it's going to require our safety or toxicity data on the whole mixture. Or if we're thinking about a component base, then it requires us to understand the individual chemicals that make up that mixture and the safety or lack thereof of those individual chemicals. We can also look at it from the standpoint of this idea of biosimilars. And the Environmental Protection Agency has talked about this in the idea of, we have data from mixture A and a lot of really good chemistry, a lot of good toxicology, and so we have now mixture B, and we look at the chemistry. Is that chemistry sufficiently similar to the chemistry that we know a good bit about the toxicology to allow us to read across and not have to repeat that toxicity information? And this is just basically what I said. Is it close enough? Are the data that we have sufficient to allow us to conclude that the toxicological significance of this new mixture, that we know the chemistry, but we don't know much about the toxicology, is it sufficiently close enough to another material that we know both the chemistry and the toxicology? And I'll show you a couple examples of that. And the goal is to use the toxicology data from one mixture to estimate the potential hazard or the risk of that mixture of concern. Now, another issue that we have to keep in mind when we think about mixtures is, typically when we do these types of studies, we tend to think additive. Compound A plus compound B plus compound B are additive. Those of us that were trained in pharmacology know that that's not always true, that we can have other types of interactions between chemicals that may or may not make our move forward a little more complex. So we need to keep that in mind. Our current approach, when we think about food mixtures, and we go to that classic book that we call the Red Book at the Food and Drug Administration, typically we do a genetic tox battery, maybe a 90-day study, and have a little bit of a feel about the exposure, and that's about where we end up with these current approaches. So we're beginning to see things coming out of the U.S. National Academy of Sciences and other places that are driving us to think a little bit more in depth about how we approach these things, and we're thinking more in terms of, can we get away from these applicable endpoints and move more towards the genomic, the other types of genomic endpoints? Can we move away from in vivo studies into more in vitro studies? And when we think about omics, we tend to think in terms of maybe transcriptomics, but there's really a lot more to that. We need to understand what's happening with all of these omics and to realize that a toxic or drug interaction is really an adverse effect on a network system. And now we're getting into very, very detailed and very difficult and large data sets that we have to work with. But if we do these systemic or systematic toxicology studies, we can begin to parcel out some of the effects to cell stress, inflammation, apoptosis, cell proliferation, DNA, just as a few examples. But that's probably a little bit beyond where we're going to be right now today with food additives. So we begin to think about searching for models that can help us do this, and we can ask ourselves the question, who has the right model? And I'm going to suggest to you that a man by the name of Wiley came up with the best model in the beginning. And I don't know how many of you are familiar with Harvey Wiley, who was at the Food and Drug Administration, and his model was what? What did he use? He used people. He used humans. We can't do that anymore for ethical reasons, but he had what he called his hygiene table or his poison squad, and he would feed his colleagues every day at lunch X amount of a chemical or an additive, because he was concerned about additives going into our food supply. And on Tuesday, 2X, Wednesday, 3X, Thursday, 4X, and so on. And if he didn't have a problem, he said it was okay. And one that he did early on was borax. Well, we don't do that anymore. So we've got to think about models, and there are just all kinds of models out there. And I'm just going to share with you two of them. One's a little worm and one's a little fish. And these are becoming tools that a lot of people are beginning to use. In the case of the fish, that's the zebrafish. It's relatively undemanding. It doesn't take up a lot of room. Its embryo develops outside the mother. It has a relatively short breeding period. It breeds throughout the year. One female can give a lot of offspring. And within a week, all the organs are developing and beginning to function, and it's transparent, so you can see these organs. So people are beginning to use this as an alternative. There's a lot of homology that exists between humans and the zebrafish, and I've just listed a few of them for you. 72% of the human genes have zebrafish. 82% of human diseases we can see within the zebrafish. And here's, we can do it for, use it for, excuse me, mechanistic studies. And here's just a very simple example. of a mixture of two materials, and we see that the mortality of the mixture increases by about 83% over the two individual components. The little worm is C. elegans, and you can see this, it's quite small. It has a lot of homology also present between the humans and between the worm. They're transgenic strains, and these are just a couple of examples where you can see that two chemicals react differently individually as when they're combined. Developmental delay also occurs with the mixture at a greater rate than it does with each of the individual chemicals, and that just shows a timeline of development between the individual chemicals and the mixture. When you compare this little worm with the rat, you can see that there's a lot of conservative in space and cost and time elements. There still needs to be a considerable validation. And if you compare the worm to the zebrafish, you can see that there's a lot of comparisons between the two. So these are a couple of examples of possible alternatives that we can use in looking at mixtures. I think the area that is rapidly developing that maybe we'll be able to apply to mixtures are what we're calling the cell or organ on a chip. And one of the big drawbacks initially is they're just individual organs, but people are now putting together. Here's an example of a lung and a liver on a chip where you can have metabolism ongoing and look at various types of cells within the lung tissue. I think those are things that we're going to be able to use as we move forward. But as we think about the issue with complex food materials, we've really got to take ourselves back into the agricultural arena and continuously remind ourselves that things can change depending on a lot of things. The species and the variety, the plant part, the agricultural conditions, and even the processing that might take place as we process the plant material. Just a very simple example, comparing a grape seed and the grape skin, and you can see that there's a fairly significant, though small, difference in the chemistry between the two. If you look at young leaf versus an old leaf, you can see the more mature leaf has developed some new compounds that may be a concern. Extraction procedure can make a very large difference excuse me, in the type of extract that you get. You can see in the case of garlic, the supercritical CO2 extraction is much more like the fresh garlic itself. So the nature and assessment is going, I think, still to require us to approach this on a case-by-case basis. And I'm going now to go through some examples to share with you, some of which I've been involved with. One example is from the national NIH, the National Toxicology Program, and another example is from Procter & Gamble. So if you look at this idea of establishing a bridge, when you think about extraction to semified pure to a purified compound, as you move closer towards that purified compound, you have less of a concern about the mixture, but now you're concerned about the purified compound. One of the problems that we continue to have with trying to compare my chemical that I know a lot of chemistry about, but not a whole lot of the safety or toxicology, is that when I go to the literature and I find all of this toxicology information, guess what? Until recently, I'm just told that it's a water extract. I don't know what that chemistry is, so I'm not able then to use that data. And we're seeing more and more in the literature that people are requiring chemical evaluation of materials before publication with the tox data. And same thing is true with purified compounds. Here's an example of the where the National Toxicology Program found themselves in, I think, a rather difficult situation. They'd done a very, very comprehensive study on ginkgo all the way through a 2-year bioassay. And then they realized that they did it on this material, but look what's out there in the market. There are just all kinds of different ginkgos. How similar are those different ginkgos? And so they undertook a program to look at this by doing two things. One, to look in detail at the chemistry, both targeted chemistry and untargeted chemistry, and some select in vitro tox studies. And when they did that, they were able, these are the numbers that they looked at. I said this is what they compared, and by combining this, they were able to show clusters. And I'm just going to show you one of their clusters. And this is a cluster of P450s in primary human hepatocytes. And you can see there is one cluster that's pretty close to that so-called gold standard, but yet there are a lot of outliers that don't fall. And their conclusion of this is that it's the untargeted chemistry and the hepatocytes from humans that gave them the best information to make predictions between all these various ginkgos that are out on the market with the one they had done this 2-year bioassay on. Here's another example that I was involved in where there was a company that wanted to bring a new pine material onto the market, and it's used for foaming in a number of carbonated drinks. But they didn't want to do the toxicology. FDA had already approved another resin, the wood resin, with its chemistry. And so what we did here was to look at the chemistry in great detail, and we were able to show that the chemistry between the two were sufficiently similar that we made the argument to FDA, and FDA bought the argument that there was no reason to do the toxicology data because the chemistry was so similar. Here's another example in which we were asked to show the safety of a material without using any animal experiments. This material has been on the market for a long, long time. That is the berry, cranberry. But it was the leaf that they wanted to make a tea from. And so what we then did was to say you need to take a very, very broad look at all of the, not just the species and the genus, but out to the family. What do we know that's potentially available from the standpoint of toxicology in this family? And grainotoxin was one of the materials that's found in certain rhododendrons that grow in parts of Turkey. And if you know your Roman history, your Greek history, when Alexander the Great went through that part of Turkey, they consumed a lot of the honey that was given to them, and it had this toxin in them, and a lot of his soldiers got sick. So you have to be concerned about things like this, although it didn't appear that it was actually in the cranberry leaf itself, but it was in that family. And so you do that, you find out what might be present, so you've got your targeted things you're looking for, and then you need to do detailed chemistry on the untargeted, and that was all done. And then you set up standard operating procedures to be sure that you don't bring in other things, that you keep out pesticides. You need to have health-based analytical lower limits. If your material is toxic at 10 parts per million and your analytical is only good to 100 parts per million, you're going to have to lower that analytical capability, and you do batch and then the final, and we were able to show that by going through with all this detailed chemistry and some very limited in vitro toxicology that this material could be used. Here's an example that Procter & Gamble did, again, looking at the overall complex of this extract, and you can see that there's only a very, very small amount of the material that's unknown, and they looked at that, and these are the chemicals. Then when they did their actual study and their fingerprinting, looking across the board, there were only 2 materials that were above this threshold of toxicological concern, a concept that was originally, I think, developed by Ian Monroe in Canada, then picked up by Pally Crow in the European Union, and used quite a bit in the European Union, that if you your material is below a certain level, you don't have to be concerned about it. And in this case, they had only 2 materials that were above this concern level, and they had sufficient data to say that there wasn't an issue there. So you can summarize all of this by really saying that it's the chemistry that you've got to delve into and understand, and if you do that, then you can look at the gaps, the supportable exposure level, and the information you need for safety studies. And this is the paper that the Procter & Gamble people published showing this, the focused approach for detecting possible bad actors in botanical extracts. But it's the chemistry that allows us to get a handle on these mixtures. The analytical chemist for years and years and years has been the toxicologist's worst enemy. They could find things that didn't exist, and then we had to explain it. Now they become our best friend in this area, helping us understand what's there so that we can move forward. Well, I want to leave you, regardless of how you decide to do your study, whether it's an epi study, whether it's a nutritional study or a tox study, with a couple of things just to think about. Inadequate training and mentoring of researchers is a critical problem in why we have so many studies that are published that cannot be reproduced. And we have to think about that. We need to think about experimental design, and we heard some of this just earlier. Things like p-hacking, that is just cherry-picking your data to find anything in my study that's positive, or hankering, where you develop a new hypothesis after your study is over because your original hypothesis didn't work for you. And then validation of your reagents. Just because it says it's something on the bottle doesn't mean that it's something in the bottle. You need to think about that. When's the last time you calibrated your instruments? Bias. Pressure to publish only positive results. We need to get the negative information out there as well. And I would just leave you with 6 red flags for suspected work by Begley. Were the studies blinded? Were all study results shown? Were all experiments reported? Were positive and negative controls? And our last speaker, she had some negative controls, some positive controls in there, and that's really great. Were reagents validated? And were statistical tests appropriate? Thank you very much.